This is an introduction to the Laboratory of Organelle Control at the Nara Institute of Science and Technology. In our laboratory, Associate Professor Yukio Kimata, Assistant Professor Yuki Kimata, and students from various countries will welcome you. In this video, you can find research projects we want to do with you. This photo shows Saccharomyces cerevisiae, a prominent yeast species. Yeasts are fungi that grow as unicellular cells. In other words, although yeasts are unicellular microorganisms, they are eukaryotes like animals and plants. Because yeasts are easy to genetically manipulate and grow, it is frequently used to learn about other eukaryotes. For example, yeast has occasionally used in researches on human diseases. Yeasts are also industrially used in producing valuable biomolecules. Since yeasts are eukaryote, it has various organelles, as well as animals and plants. The endoplasmic reticulum, or the ER in short, exists in the cell as flat or tubular sacs. In yeast cells, the ER forms a layer around the periphery of the cell, and the nuclear envelope that covers genomic DNA is also part of the ER. Therefore, when fluorescent proteins localized to the ER are expressed to illuminate the ER, it appears as a double ring, as shown in the photo on the left. The main role of the ER is to serve as the place where proteins secreted outside the cell are folded and modified. Many proteins, such as cell wall proteins and pheromone proteins, mature in the ER and are transported out. In addition, various lipid molecules are biosynthesized on the ER. The aim of our research is to elucidate the mechanisms by which living organisms adapt to environmental changes, namely stress tolerance, using yeast cells as a model organism. Furthermore, we seek to connect these findings to industrial applications. We are particularly interested in ER stress. Under normal conditions, peptides produced on the ER membrane are folded inside the ER to become mature proteins. They are then packed into transport vesicles, pass through the Golgi apparatus, and are finally delivered to the cell surface or other organelles. However, as shown in the picture on the right, when the cell's environment worsens, protein folding in the ER doesn't progress efficiently, and abnormal proteins accumulate inside the ER as unfolded proteins. This state is called ER stress, and in some cases, leads to cell death. The phenomenon that protects cells from ER stress is called the unfolded protein response, or UPR in short. UPR is triggered by ER stress and increases the expression levels of hundreds of genes. Then, the ER becomes more active and expands in size, leading to recovery from ER stress. ER stress occurs in various situations. One is ethanol. When cells are exposed to ethanol at high concentrations, ER stress is triggered in both animals and yeasts. In addition, in plants and yeasts, high temperatures are also known to lead to ER stress, likely because of impaired protein folding at high temperatures. Furthermore, in animals, ER stress is linked to various diseases, including virus infection, neurodegenerative diseases, cancer, and diabetes. One of the research topics that our laboratory is going to be engaged is the relationship between ethanol fermentation and ER stress. Saccharomyces yeasts are famous for producing ethanol from sugars, namely ethanol fermentation. This process is commercially and industrially used for brewing of alcoholic beverages, bread making, and bioethanol production. On the other hand, we have discovered that, in yeast, ethanol induces ER stress and triggers the UPR. However, it is still unclear when ER stress is induced upon during actual ethanol fermentation, and how the UPR affects ethanol fermentation. By approaching these questions, we believe we can contribute to improving ethanol production by yeast. We are also engaged in research projects using yeast species other than Saccharomyces. While most yeasts can grow at temperatures around 30 degrees Celsius, Cluiveromyces marxianus possesses a remarkably unique characteristic to grow even at temperatures close to 50 degrees Celsius. Because high temperatures are known to induce ER stress, we are investigating the role of UPR in the growth of this yeast species under high temperature conditions. We will also try to find factors for heat tolerance of this yeast species other than the UPR. Additionally, this yeast species has a high capacity to use sugars derived from cellulose, and it is promising for biofuel production. In collaboration with Chulalongkorn University in Thailand, we are also aiming to develop strains showing high tolerance to alcohol stress, toward high production of bioalcohol by this yeast species. We are also engaged in fundamental molecular and cell biological research using Saccharomyces yeasts. In the yeast UPR, IR1, an ER-located transmembrane protein, acts as a sensor for ER stress. When cells are not stressed, IR1 is distributed throughout the ER. However, under ER stress conditions, IR1 molecules cluster together to form large oligomers. This results in the production of the transcription factors HAC1, HAC1, which transcriptionally induces a large number of genes, leading to activation and enlargement of the ER, thereby alleviating ER stress. However, it is still unclear how IR1 sense a wide variety of stress stimuli, and since the expression of hundreds of genes is altered by HAC1, it is also still unclear how it affects to various cellular functions. We are now working to answer these questions. Our basic biological study expands to the research to investigate what happens when toxic proteins that cause human diseases are expressed in yeast cells. Human polyglutamine diseases are caused by genetic mutations that insert a long stretch of glutamine residues into a protein. One example of polyglutamine diseases is Huntington's disease, which leads to dysfunction of motor neurons and movement disorder. GFP attached a large number of glutamine residues, namely polyglutamine GFP, is known as a model of toxic proteins inducing polyglutamine diseases. When expressed in yeast cells, as well as in human neuron, polyglutamine GFP aggregates and exhibits dot-like distribution. Although these aggregates form outside the ER, they induce ER stress. Then, our study will aim to clarify the molecular basis underlying this toxic effect of polyglutamine GFP. In a project toward application of Saccharomyces yeast, we have created cells in which the UPR is artificially and constitutively induced. In nature, ER stress produces the transcription factor HAC1 to trigger UPR. Then, what happens when HAC1 is artificially and constitutively expressed using a constitutive gene expression promoter? In this slide, cells were cultured under non-stress conditions, and the ER was observed by an ER-located fluorescent protein. As shown in the left panel, in wild-type cells, the ER appeared as double rings of the peripheral ER and nuclear envelope. In contrast, as shown in the right panel, In cells artificially expressing HAC1 using a constitutive gene expression promoter, namely, in constitutively HAC1 expressing cells, the ER was expanded and spread throughout the entire cells. Because the size and function of the ER are enhanced in the constitutively HAC1 expressing cells, they can highly produce heterologous biomolecules made in the ER. One of such heterologous biomolecules is beta-carotene, which is a yellow or red-colored pigment originally made in some organisms, such as carrots. This photo shows an agar plate on which yeast cells have been grown. Because normally, Saccharomyces yeast cells do not produce pigments, they exhibited white color, as shown in the top two samples. In contrast, as shown in the bottom two samples, they were colored when introduced beta-carotene biosynthesis genes obtained from another organism. Importantly, because the ER is activated in the constitutively HAC1 expressing cells, they produced more beta-carotene and exhibited darker color than wild-type cells. By introducing genes from other organisms into yeast cells, it is today possible to produce various commercially valuable heterologous biomolecules by yeast cells. Human secretory proteins, such as antibodies, cytokines, and insulin, can be used as pharmaceuticals. Moreover, in the near future, it may be possible to commercially obtain lipid molecules from genetically modified yeasts. Because they are produced in the ER, we expect that activation and expansion of the ER can increase their productivity. In this viewpoint, we will further improve our strategy to artificially and constitutively induce UPR, for instance through combination with other techniques. In summary, we investigate cellular functions related to the ER in yeast cells from the viewpoints of both basic and applied sciences. We are happy if you can join our team. Moreover, any questions are welcome.